Sterilization and disinfection. These are the important two terms which are commonly used in biosafety. अब हम देखेंगे कि स्टरलाइजेशन और डिसइंफेक्शन में मेजर क्या डिफरेंस है. Now sterilization is a process by which we can kill all types of microorganisms including the bacterial spores. जबकि डिसइंफेक्शन में हम बहुत उन माइक्रो ऑर्गेनिजम्स को किल कर सकते हैं जो कि इन्फेक्शियस हैं, बट नॉट ऑल टाइप्स ऑफ माइक्रो ऑर्गेनिजम्स. इसके अलावा डिसइंफेक्शन के ज़रिए हम बैक्टीरियल स्पोर्स को भी नहीं मार सकते. सो दिस इज़ द मेजर डिफरेंस बिटवीन स्टरलाइजेशन एंड डिसइंफेक्शन, दैट डिसइंफेक्शन कैन किल ऑल सॉर्ट ऑफ माइक्रो ऑर्गेनिजम्स while disinfection is suitable for only few microorganisms. So disinfectants kill some microorganisms, not their bacterial spores. Now, disinfectant are the chemicals which are used in the process of disinfection, and they kill many microbes, but they cannot kill all type of microbes, not even the bacterial spores. इसके अलावा term antiseptics use होता है. These are also the chemicals, mild form of chemicals used for the process of disinfection. Now let's look at the methods that by following them we can go for sterilization and disinfection. There are two major ways by which we can sterilize the apparatus, the solutions or anything else. Now the first is the physical agent and the second one are the chemical agents. Physical agents include heat, which we further subcategorize them into moist heat and dry heat. The second is radiation. Usually we talk about the UV radiation used for sterilization purposes. Then filtration. Here we use special filter papers containing small pore size that not only kept the microorganisms away, but also small, very small size microorganisms such as viruses. Now we also use the process of filtration for those substances that get destroyed by heat or by UV radiation. For example, enzyme solution और sugar solution को हम heat के ज़रिए sterilize नहीं कर सकते, क्योंकि इसके ज़रिए ये खदशा है कि enzyme या sugar solutions destroy हो जाएंगे. For that we use the process of filtration. Then the second method used for sterilization and disinfection, these are the chemical agents. There is a list of chemical agents, but routinely for surgical apparatus we use glutaraldehyde because it is the best chemical agent for sterilization. Apart from this, we can also use formaldehyde for histopathological sections. Now, if you want to make our water sterilize or disinfect, we can use chlorine, and the process is known as chlorination. Now let's talk about the physical agent, which is heat. As I've told you that we have divided heat into two groups, dry heat and moist heat. So first let's talk about the moist heat. हम पानी घर में sterilize करते हैं. How? By boiling. Then steaming is another way of moist heat sterilization. लैब्स में ऑटोक्लेव अपरेटर्स होता है जिसमें हम अपने ग्लास अपरेटर्स को सॉल्यूशंस को स्टरलाइज कर सकते हैं at 121 degree centigrade. Pressure should be of 15 pounds and leave the apparatus or the solution in the autoclave at this temperature and pressure for 15 to 20 minutes. Then pasteurization is a process by which we can sterilize the milk below 100 degree centigrade, क्योंकि हमने मिल्क से सारे माइक्रो ऑर्गेनिजम्स को डिस्ट्रॉय भी करना है और उसके कॉन्स्टिट्यूएंट्स को मेंटेन भी रखना है. So for the sterilization of milk, we use the process of pasteurization. Then the second form of heat used for sterilization is dry heat, such as red heat. If we are using inoculating loop in a microbiological lab, then you have to sterilize the inoculating loop before inoculation. So simply place the inoculating loop in a blue part of the flame, and when it gets red hot, it is sterilized. Then flaming. For example, if you are using any sort of forceps, glass rod, First dip it in a spirit and then pass it through the flame for sterilization. Then thirdly, you can sterilize glass apparatus by placing it in a hot air oven at 80 degrees centigrade. Now never try to sterilize gloves by heat because there are some substances including gloves that can catch fire. Then the second physical agent after heat is UV radiation. Here we use simple UV lamps for sterilization. Now these UV radiations can penetrate the cell wall. They simply disrupt the genetic material. As a result of this, the cell is unable to reproduce. Now UV radiation is commonly used because it is UV lamps. Lamps are easily available, and after sterilization no toxic residues are produced. Now some time is required for heat sterilization or for any other form of sterilization, but if we are using UV radiation, we require short contact time. Then equipment, that is UV lamp, is easily available, easy to operate, and even it is easy to maintain. So these are the different methods which are used for the process of sterilization and disinfection.